Good morning, everyone. My name is Yasmin Rahimi. I'm a senior research analyst at Roth Capital Partners. It's really my pleasure to introduce you with a great set of panelists. The title of our panel is Metabolic Disease, Fibrosis, and NASH. Our focus is on lessons learned from NASH development in 2019 that will shape 2020. The format is, for the next 40 minutes, 45 minutes will be questions asked by me, and then I'll open it up to you. You're more than be happy if you want to, stop us in between or and just raise your hand. We have a mic, and then we'll allow you to. I know it's pretty early. Everybody's exhausted, so we want to make it very interactive as possible. So, with that said, dear panelists, everyone, Bernard, Robert, Tom, Ken, thank you for being here. Starting with Ken, please introduce yourself, maybe state briefly one lesson that you've learned from your own NASH drug development in 2019, and maybe one lesson from sort of the macro NASH environment.
So I'm Ken. I'm the CEO over at a company called Metabolic. We're a biotech company working on oral small molecules for NASH as well as other conditions. I'm actually a gastroenterologist, hepatologist by training. I left medicine 13 years ago and have been in sort of venture capital and industry since. So lesson learned from 2019. Most everything's going to fail. This is a very difficult disease area. I think people are overly optimistic on their mechanism. I think the reality is we're going to see way more failures still to come in 2020 rather than successes because this is a difficult area to treat. And then the macro side of things, from an investor perspective, I think it's been interesting to see the public markets and how they've reacted to companies. I think there was an over-exuberance in NASH in 2018, where if you were a company and said that you had something that was NASH, then all of a sudden investors would give you money. And then last year, if you said you were working on NASH, investors were running away from you. So I think if you just take a step back, this is clearly a clinical problem where the magnitude and the number of patients is in the many millions. And while there's no yet approved therapy, and we're on the verge of possibly seeing the first one, this will definitely be a market that I would not bet against. But it's going to be really hard to actually develop drugs that are going to make an impact.
You can. Thomas?
So good morning, everyone. My name is Thomas. I've been with Novo Nordisk now for 17 years in this role for a decade or so. I've been following the NASH field very, very closely and kicked off our scouting and BD activities, about 5 years ago in NASH, to see it evolve from this very pleiotropic approach and many I think we counted 56 different mechanisms of actions in the pipeline 5 years ago. It's bound to fail. But that's really learnings, and that's the biggest learning I have from 2019 is that we need to really make use of the failures and not repeat the same mistakes again and again. I think industry really should get together and discuss why did it fail? What was the mechanism of action that worked and not worked? And we're not good enough in sharing those experiences. That's the biggest learning for 2019, I think. Our appetite is still sky high. Of course, we didn't fail in 2019. May 2020 will be when we may fail or be successful with exploring GLP-1 in NASH patients, of course. So my appetite right now is very much tuned to May event and decisions made after the readout from that.
Great. Good morning, everyone. I'm Robert Jackson, the CEO of Indolo Therapeutics. We're developing small molecule integrin antagonists for fibrosis. So I guess my perspective, I've been with the company only about 14 months, and before that I had no background in fibrosis or NASH, so it's been interesting coming in. And I guess my perspective is half glass empty and half glass full. And that is that I certainly concur that I think this feels a lot more difficult than some people thought it was going to be a couple of years ago. Just the mass complexity of the disease, the difficulty in diagnosing the disease and staging patients and measuring outcomes, and I think is incredibly difficult. But I think the glass half full is that that offers opportunity. The patients aren't going away, has been mentioned, but also all of these different aspects of the disease mean there's not going to be a single answer for the disease either. So I think there's a lot of opportunity for a lot of different products and interventions to come in for different patient types, different stages, and different combinations. And so I think there's going to be a lot of opportunity there, even though it's going to take longer than some people thought.
Good morning, everybody. My name is Bernard Cooley. I'm the CEO of Pliant Therapeutics. We are a local company here in South San Francisco, have been around since 2015. We are really a kind of a fibrosis play in general. We focus on a number of diseases and organs, including liver, of course, lung, kidney, muscle. I'm a gastroenterologist as well, but have been out of practice since, I think, 20 years. So I'm not going to kind of mingle with any clinical interventions at this point in time. Our programs are also, as Indelo, focusing on integrins as a therapeutic platform for fibrosis because they play a key role in activating the master switch of fibrosis, which is TGF-beta. I think in terms of learnings from our own development and also from kind of the macro level, I think a little bit more in detail, we should focus on fibrosis. I mean, we did a deal with Novartis, and we will come back to that. But I think fibrosis is one of the key drivers in NASH that determines kind of almost independently as a predictor of outcome, so of bad clinical outcome. If we look at the current drugs that are in development, their antifibrotic effect is modest at best, and I think we should focus in trying to improve on that. Because also that's where you will align with payers. I mean, pricing will be an issue. I mean, this is we assume that the Intercept drug will be approved. Pricing will be key, and will, I think, define also kind of the midterm future of this field. If it's too expensive, it's not going to for I would say kind of a mid-level medical need of disease, it's not going to fly. So we really have to focus on fibrosis, which is the end stage of NASH.
Thank you, Bernard. That sort of leads me to my next question. As many of you are aware, the two key regulatory endpoints for NASH development in phase 2b and in pivotal are NASH resolution without worsening of fibrosis and 1-point improvement of fibrosis. So I guess for my panelists, the question is, which of these two endpoints matters most to hepatologists, endocrinologists, and cardiologists? When we speak with hepatologists, we tend to have, everybody has sort of their own flavor and thoughts, but I would love to hear your comments around which one you think matters most and maybe one quick reason why. Thomas, I'm going to have you start first.
So I'm really not the expert in commenting upon that, but so nevertheless, both are important, and I think we don't have data enough to discriminate between them. But clearly, fibrosis, I echo what Bernard says. That's what we focus on, and resolution of fibrosis is going to be difficult. So that's all I can comment upon. So I think both are equally valid from that point of view.
Ken?
I was just thinking about your question. I mean, it's tricky, right? Because if you look at the liver and other conditions that we've seen, let's go back to Hep C. If you were to ask a gastroenterologist or a hepatologist 20 years ago, what's more important? Is it important to reverse the cirrhosis in a Hep C patient, or is it more important to get rid of the Hep C? Both of them are important, but the ultimate solution was to get rid of the hepatitis C, because once you do that, then you obviously prevent the progression of fibrosis and the development of sequelae. In the situation of NASH, I think we don't understand what drives fibrosis in these patients. People don't even understand why, if you have a fatty liver, do some progress to NASH, whereas others just stay benign. The data out there, at least what's been clinically presented thus far, suggests that a reversal of fibrosis is most likely to correlate with clinical outcomes. Because actually, a fibrosis reduction of 1 point or NASH resolution, that doesn't really change. We don't know whether or not that ultimately changed the outcome for the patients from a cardiovascular outcome, mortality, overall mortality, et cetera. I think the evidence would suggest that you do want to make an impact on fibrosis, but I'm also highly skeptical of purely antifibrotic therapies as monotherapy, because I actually to maybe be a little provocative, I don't think that's going to ever work. I think they need to be used in combination with a metabolic agent. And I sort of use this analogy of if you can't take away the insult, the inciting insult, which is probably toxic fatty acids, as much as you're an antifibrotic, you're always just going to be trying to catch up. So if you have a bathtub full of water and you've got the faucet turned on at full speed, that's sort of like your toxic lipids, right? Your antifibrotic might be the drain, and you could pull open the drain, but the problem is you might decrease the flow of the filling of the bathtub, but as long as the water's still on, you're always never going to be able to sort of cure it. So I think there is a promise for antifibrotics, but not as monotherapies. I don't know that, aside from minor improvements in IPF, everything else as an antifibrotic has not worked. But I think in combination in the liver in particular, it can be effective.
Thank you, Ken. Yeah, Bernard.
Yeah, I fully agree with Ken. I think we have to see this in combination therapy. Also, if your antifibrotic works and you bring it back, there is still NASH, right? So that kind of may increase. So you have to control both processes. The challenge for companies like ours is that, first, there are no approved drugs yet. So combination therapy in clinical development is impossible unless you have both assets in your pipeline. And that, by definition, is a big pharma play. So I think for smaller companies as ours, partnerships are key in trying to develop your own drug in combination with an existing, be it in development, NASH type of drug.
Robert.
Yeah, I mean, I certainly I agree with what others have said here. I think I would even take it a step further. I mean, we're kind of our vision is very siloed and narrow, and here are drugs, but then we think, oh, it's really about liver health, and so it's a combination, but it's not really. It's really about patient health, and so I mean, these patients, the reason they have this disease is because they have other health problems, and so it actually doesn't even help us if we help their liver disease if they die of something else. So I think it's really going beyond just is it the fibrosis, is it the steatosis and the cause of the insult, but what's causing that? And so I mean, we're playing our parts, but I think we do have to think about how does our part fit into that whole picture.
And I have to comment up on this because that's exactly the problem. We don't understand the etiology of our patient population here in NASH. And what we see is a very heterogeneous patient population and background, and also obesity, diabetes is certainly playing a role, but we don't know how to really differentiate between these patients in the background. So it's going to be interesting to see the PPARs, and if we can solve insulin resistance, that might actually help quite a lot in NASH patients as well.
Thank you. So 2020 is a big year for the NASH space with a number of readouts across phase 3 and phase 2s. Maybe the first question for you guys for the audience is to quickly tell them your current NASH program, your lead asset, one sentence, its mechanism of action and why it makes sense in NASH, and maybe any catalyst that is set for readout in the next 12 months. And then a part 3 is, which data set are you most excited about to see in the next 12 months besides your own? Okay. I'm gonna I'll start off mine. I know a lot of my companies, I cover many of the NASH space. I actually cannot wait to see Tomas' study from semaglutide, so that I'll kick it off like that. Ken, you wanna get started?
Sure. So we have several programs that we're working on at the company. We're working on FXR, which I think people are familiar with because that's what Intercept has with OCA. We have an SSAO program, which is an anti-inflammatory. And then we're also working on a genetic target called HSD17 beta 13. These are all oral small molecule programs. Our FXR program, maybe as early as next week, we'll release top-line data from a 12-week study that we just completed. When we ventured into this several years ago, I actually thought we were crazy for doing this because there were so many other FXR agonists out there. But we developed a hypothesis as to what we thought was best in class and why we thought Big Pharma got it completely wrong. And I think our data will prove that that's actually correct. So I think just stay tuned for that. I think that'll be I think that will be well received when we announce the top line next week. And in terms of most anticipated data, I would say I am very interested in what Novo has to present with the GLP-1s, because the weight loss has been shown, at least through bariatric studies and other things, to be associated with significant improvements. And I think there's a lot of anticipation and a lot of read-through, not only to Novo's program, but many other programs that have GLP-1 as part of the therapy.
Thank you, Ken. Tomas?
So of course, being again May 2020, we have it. I don't think we publicly said exactly the date for the decision of phase 3, stop, go, no go. It's going to be really fascinating, and we hope that the data is going to be clear enough. It will be obesity, weight loss driven. It's clear. Will it have any effect on inflammation in fibrosis? I mean, mechanisms of action of GLPs in NASH have really not been studied well enough to say that, but there is a possibility. It will very much guide me on my appetite, or our appetite on assets out there. Right now, I'm looking at, or we are looking at, preclinical and phase 1, early phase 1 assets. Depending on the readout on SERMA NASH, I might be interested, or we might be interested. I have to change that attitude. We might be interested in phase 2 and phase 3 assets. And of course, as you all know, we are looking and have already established collaborations in combos, because I think that's the key.
Great. So, our lead agent is an integrin antagonist of alpha V beta 1, 3, and 6. And we might get into later why we think those are important targets. But more at a higher level, the mechanism of action of our drug is a direct antifibrotic. And what I mean by that is that we actually have no effect on steatosis or inflammation. And we can show that in vivo. And I think the reason that's important is Thomas mentioned, I think there were 56 mechanisms of action in development for NASH. It seems a very crowded field. But I would think probably 55 of those have some impact on steatosis or inflammation, whereas direct antifibrotics, integrin antagonists like appliance compound like ours do not. I would guess you just don't either. I haven't seen the data. So the reason that's important is in the world of combination therapy or sequential therapy, like we're talking about, looking for agents that have different mechanisms of action, I think would be important. So I think that's how I'd position our compound. As far as the stage, so our lead compound has finished healthy volunteer clinical trials. We're currently enrolling in a phase 1b proof of biology trial in IPF patients. And we're taking a little bit different approach to some. We're studying our lead agent in both liver and lung, and then we'll choose, based on the data and the science, the best indication for late-stage development. But the lung is a really interesting place to start because it's a good, you can semi-invasively access the organ, and it's been shown to be very responsive to certain biomarker movements. So it's a good proving ground, we think, to prove the biology of the agent.
So our lead asset is also an integrin inhibitor of 2 integrins, and it's not in NASH. So that's really kind of the key driver and the key focus of the company right now. The lead indications are IPF and primary sclerosing cholangitis, because that's a strategy of the company focusing on orphan fibrotic diseases. We are enrolling a large phase 2A. It's a global trial. It's a 3-month trial. Readouts will be early 2021. As for our NASH asset, which is our second program, it's a selective alpha V beta 1 integrin inhibitor, and we can come back to the mechanism of action. But basically, these kind of integrins, including Indul's compound, are the activators of TGF-beta. And TGF-beta is the master switch of fibrosis in any kind of organ, including in liver. And we have evidence that this specific receptor is upregulated in function of stage of the disease with the highest levels of expression in F4. This is based on biopsy work. And that we also see TGF-beta activity being the highest in those end-stage patients. From a clinical data set point of view, the compound, we filed the IND. We anticipate to start a phase 1 for this NASH compound in February. And so, the readout will be somewhere mid-2020. Of course, this is a partnered compound, so Novartis owns the compound as we speak, and they will take further development phase 2 and beyond on there. So in terms of what I think will be critical are two things: safety and tolerability. Of course, it has to work, but if you look at some of the events that happened last year, notably with the PPARs, with CymaBay hitting a safety issue, I think, despite the fact that end-stage NASH is a serious disease, the tolerability for safety signals is very low. Right? It has to be safe. I think from our perspective, what will be key is, as everybody kind of already alluded to, this will be in combination therapy. So that means that you have a mode of action. You need to have a mode of action that it can be combined with something else without running into safety issues, that you don't have drug-drug interaction potential. And so those are like the nitty-gritty of drug development, but key when you talk about combination therapy.
Thank you. We're going to dive now sort of in very specific questions for each of the programs, starting with you, Ken. At AASLD, data was presented, interim results from 28 days, non-invasive biomarker from MET409, a sustained non-bile acid FXR agonist in NASH patients. Can you enlighten us a little bit about the chemistry, why sustained versus non-sustained matters in mitigating safety concerns? And then also for our audience who isn't familiar, what are key biomarkers that predict target engagement?
Sure. So FXR is a nuclear hormone receptor, the farnesoid X receptor, that's been implicated in regulating a whole host of different downstream pathways and activities, best recognized for its ability to regulate bile acid metabolism. And the reason that FXR, I think, is really probably the most interesting target in NASH, at least as of today, is because there's a company, Intercept Pharmaceuticals, that's on the verge of FDA approval for their FXR agonist, which is a bile acid. When we started our program several years ago, I think I mentioned this earlier, there was a multitude of others in this space that had competitive programs that were much further along than ours. Gilead, Novartis, Enanta, et cetera. And I think for us, as we started to venture into this space, we wondered, why should we, as an emerging biotech, work on a target that seems to be overly crowded? And there were two things that emerged that we hypothesized that could give us a best-in-class profile. The first one was actually biology, and it was to actually engage with the target, FXR, on a continual basis, which is what we call sustained activation. That's a terminology that we introduced into the industry, whereas many of the other non-bile acid FXR agonists, such as those from Gilead and Novartis, et cetera, are transient, meaning that they only engage with the receptor for part of the day. We had generated preclinical data to suggest that that was going to actually sacrifice efficacy, and therefore that was one of the design features that we decided to employ, was to actually have a sustained FXR agonist to maximize safety. In addition to that, we started on a novel chemical scaffold. So our starting point is an old molecule called fexaramine that's chemically very distinct than all the other FXR agonists that are in development, including those that have historically been developed. And that is just a different chemical structure that interfaces or interacts with the receptor probably slightly differently. And given the multitude of effects that FXR has as a nuclear hormone receptor, we think that ultimately translates into differential downstream effects. We did present data last year from a very, very short single-center open-label study where at 4 weeks of treatment with a low dose, we were already able to demonstrate a 20% liver fat reduction. And we just completed a 12-week study that will look at a couple of different doses in a randomized placebo-controlled trial. Again, the top-line results of which we plan to release probably in the next week or two. And I think it's basically around having that sustained profile and having the novel underlying chemical structure that I think gives us a much wider therapeutic index than obeticholic acid, which is on the verge of approval, as well as other FXR agonists in development. I think what's interesting, at least from my view, is that while the field appeared to be very crowded 3+ years ago in FXR, the only competitor that we really see is OCA as a viable FXR agonist in the field. But they have limitations in terms of magnitude of efficacy and side effects, and that's where we think we can differentiate.
Thank you. Tomas, we're really, looking forward to the data in May. There's enormous amount of significant data existing that if you lose more than 10% body weight, you see phenomenal effects on NASH resolution as well as fibrosis. Maybe the response that you see in NASH resolution tends to be more significant than you see with a fibrosis. I would love to hear your thoughts on sort of what evidence do we currently have on does semaglutide has a direct anti-fibrotic effect that maybe we're not aware of? And what are your thoughts on weight loss and fibrotic effects, just because it doesn't seem as clear as improvements in NASH resolution?
So, as I mentioned, we expect it's weight loss driven resolution of liver fat NASH. There are early indications in the literature that GLP-1 has some anti-inflammatory effects. I wouldn't take, we have not counted on that. That's very clear. And we do not expect, if it's a very positive upside if that holds true in this study. If you look at the diabetes studies that we've done, it's been very difficult to prove any anti-inflammatory effects. That's clear. But if you lower your blood glucose and you're well controlled, of course you will have a whole body which is responding much better to other interventions as well. And I think that's really the key. Coming back again, we do not expect antifibrotic, any large effects in this. And therefore we're looking for combinations. And I talked with other panelists, and it's a little bit sad that the industry failed in 2019 and investors are shying away, as we heard, or not shying away. We are not. I have been in really deep discussions with Bernard, Robert, and Ken. We didn't manage to get to a signature, but extremely deep discussions. And that is just showing our appetite. So I think Big Pharma in the play of NASH is definitely very interested still. I just want to emphasize that so that there is a buyer still out there.
Very good. We will summarize that in our note so that everybody makes sure that they saw this comment and if all the stocks are moving up on it. Thank you. We need that.
Well, just to follow up on that, I would say, based upon our meetings that we're having at this conference, that there is a lot of interest in NASH from the large strategics, not just from Novo Nordisk, but from others. And I think that 2019 was I like to think we sort of hit bottom. And just judging by the fact that there's so many people in this room, clearly there's an interest also, right? So I think that's all a good sign.
Thank you. Robert, thank you for being here. We recently saw preclinical data also at ASLD from IDL-2965, an inhibitor of alpha V beta 1, alpha V beta 3, and alpha V beta 6. So in November, can you enlighten our audience to the rationale for selecting these particular isoforms for the treatment of NASH, and then maybe a little bit more color about which tissues besides the liver express alpha V beta 1, alpha V beta 3, and alpha V beta 6, and maybe the selectivity versus in the liver versus other tissues?
Yeah, thank you. So, we can show upregulation of all three of those targets in liver fibrosis, in lung fibrosis. We're building out that science in kidney fibrosis. We can show empirically that our compounds are very active in kidney fibrosis, as well as cardiac fibrosis, skeletal muscle tissue, pancreatic. It's the mechanism seems to be widely applicable. And specifically, our lead asset, alpha beta 136, alpha beta 1 and 6 are involved in TGF-beta activation. As Bernard mentioned, that's a master regulator in fibrosis and a really important mechanism. But integrins are involved in other aspects of fibrosis beyond just TGF-beta activation. And so alpha beta 3 is a really important part of our story because alpha beta 3 is not involved in TGF-beta activation, but it's involved in some functions called matrix sensing functions by which fibroblasts can migrate to the sites of fibrosis and then have a survival signal and avoid undergoing apoptosis. And so bringing in alpha beta 3 gives our compounds, we believe, very, very potent antifibrotic activity and broad utility across different organ systems. And so it also allows us not to have to hit so hard any one target and rely on just one target and one mechanism for efficacy, not have to drive that down so far. So that's the overall. I'm not sure. I can't remember what the other part of your question was.
No, that was perfect.
Great. Okay, thank
you. That's great. Bernard, congrats for securing a large pharma partner with Novartis, and plus with the largest preclinical NASH upfront payment of $80 million. Can you briefly walk our audience through, who are not familiar with Pliant, through the integrin discovery platform and its uniqueness, and give us a glimpse of what percentage of the 6,000 plus compounds in the library would be ideal for NASH, and then second, explain sort of the advantages of targeting fibrosis. Sorry, my questions are so long, I know. I was writing them on the plane and I had no access to, couldn't get on the Wi-Fi, so I just kept making them longer and longer. I had nothing else to do. I couldn't get, so that's why.
No, thanks. Yeah. So, I mean, the deal that we did with Novartis was around the NASH compound. As you said, it's to our knowledge the largest preclinical, largest upfront for a preclinical asset so far. But to your point, we actually have a whole integrin platform behind that, and that's also reflected in the deal with Novartis because they brought 3 additional integrins to the table, and that's also mentioned in the press release, so that's public. And so we will screen against those, and we have our internal programs as well, which are earlier stage. So the integrin platform is really serving the ultimate goal of the company, which is bringing compounds to fibrotic diseases, notably focusing on some of the orphan fibrotic diseases. We think that the integrin target class as such has multiple ways of interacting with fibrosis. And I think to Robert's point, one is TGF-beta activation and inhibition thereof, being the master switch of fibrosis, but also looking at other integrins like collagen receptors or activators of myofibroblasts independent of TGF-beta, which will allow us to kind of do combination therapy, or at least have a combined approach to fibrosis as such, which remains a very difficult issue. So coming back to the platform itself, we have built an integrin-focused library of close to 8,000 compounds now, initially coming out of our screening campaign for the lead asset. We are a spin-out of UCSF, and a lot of the work we do was based on the biology research that Dean Shepard did, and then Bill DeGrotto, who's a chemist on faculty at UCSF, is one of the leading integrin chemists in the world. So their work led basically to what we have built in terms of platform. Now, you can't build an integrin-focused library without having a screening system. So we have a high-throughput screening system, invested a lot of money in that, allowing us to screen hundreds of compounds on a weekly basis against, I would say, most of the 24 integrins. And all of that feeds into a biology platform that is focusing on understanding how these compounds interact with integrins, different conformations, etc., and then at the same time feeding into our fibrosis drug discovery and drug development engine, which includes advanced cellular assays as well as, of course, the animal models, but notably human tissue. So a lot of the work we do is on fresh human tissue from patients undergoing transplant for liver or lung, and now we are expanding indications. These are like end-stage disease types of tissues, allowing us to evaluate our compounds and competitor compounds in terms of their effect on profibrotic genes. So the second part of your question is, what's the advantage of using an integrin to selectively block activation of TGF-beta versus pan-TGF-beta inhibition? I think we know that pan-TGF-beta inhibition is probably one of the most anti-fibrotic ways to treat patients. But the problem is, and we know that from the oncology space, where TGF-beta receptor antibodies have been tested, that the systemic toxicities are tremendous. So there's cardiotoxicity, neurotoxicity, it's pro-inflammatory, potentially pro-cancerous. So it's just, there's too much toxicity for some of these, like, liver fibrosis or lung fibrosis type of patients. So the only way to address it is to find a tissue-specific, fibrotic tissue-specific manner to inhibit TGF-beta, and the solution to that are these specific integrins like alpha-V-beta-1 and alpha-V-beta-6, which are components of our lead compounds, both for Indelo and ourselves, which are solely expressed in fibrotic tissue. Their expression levels in other tissues are extremely low or even not there.
Thank you. Another hot topic currently in the NASH drug development is drug reimbursement. So do you believe FDA and payers are likely to require biopsy-proven NASH for the first batch of approved NASH drugs, especially if the price tag is high? And if it is the U.S. healthcare system equipped to demand the need of biopsies?
I'll start. I'd be surprised if the FDA requires a biopsy-proven NASH. It would be unprecedented. I mean, I could be wrong here. I haven't taken a drug through the regulatory pathway, but just trying to use common sense, I think it would be completely illogical to have that in the label. It's not like they say you need to have biopsy-proven colon cancer or biopsy-proven breast cancer, et cetera. I don't think it's the FDA's job to dictate to clinicians how they diagnose or don't diagnose a disease, clearly because that's always sort of a moving target. I think with the payers, it's a different story. I think that the payers, I would not be surprised if some payers do require that, just because having actually taken a diagnostic product all the way through full reimbursement, payers get very nervous when they see millions of patients that could potentially be placed on any therapy or be given a diagnostic. And I think because of that, the last thing they want to do is be surprised with a huge flood of claims or reimbursement that they need to pay out. So I think if we're going to see it anywhere, it's going to be amongst the payers, but I'm hoping that the FDA won't break precedent and try to include that in the label, which would be odd to me. I'm interested to see what other people think.
I mean, we are part of the Liver Forum, and that's one of the discussions that are taking place there. And basically, it's unlikely. I mean, you never know, but it is not anticipated that language like treatment of liver fibrosis in biopsy-proven NASH is going to be included in a label. I think now taking the fibrosis angle again here, I think one of the advantages of going to F3, F4 is that you don't need a biopsy to kind of separate out those patients. In principle, imaging or some of the new serum markers will help to kind of diagnose those advanced stage liver fibrosis patients versus F1, F2 type of patients. So you don't even need a biopsy to kind of prove the disease there.
Tomas and Robert, would you like any comments?
I have exactly the same comments, and I echo.
Okay. So I would like to move on to a fun session. The questions are, would you rather? Each panelist will be asked, would you rather question, and they must state their answer and the rationale for selecting that answer. I figured it would wake people up, right? Sounds fun. Let's give it a try. We'll start. I'll go in a different order. I'll ask Robert first. Would you rather partner IDL-2965 sooner or later in clinical development? Please explain your answer. I know these are kind of silly, tough questions.
Well, I'm going to be uncooperative and say that we see a partnership as a means to an end, not an end, right? So ends, and these are not the only ends, but ends are getting our drug to patients and helping the most patients as possible. Ends are getting a fair return for my investors and the like. So I think that's the lens through which we look at potential partnerships. Is it going to help us to advance our asset in ways that we can't do ourselves? And is it going to be getting a return for my investors, which is something we need to be considering. So that's how I'm going to answer your questions. So we evaluate each in that way.
I shouldn't have started with you. You ruined it. Yeah. So okay, I'm going to pick Ken next. Ken, would you rather combine an FXR agonist with a thyroid beta agonist or GLP-1? And don't pick GLP-1 just because Tomas is sitting next to you and you don't want to upset him. Be honest. Explain your answer.
I don't think it has to be mutually exclusive. So I think we'd be open to partnering with all different modes of action. I think for us, we haven't yet partnered with anyone just because we haven't had clinical data. And I think that there's skepticism out there as to whether or not anyone can really have a differentiated FXR. And so again, I know I keep on saying this, but I'd say once you sort of see the top line data from next week, I think there'll be a very different sentiment amongst those in the industry because I think we'll show things that were completely unexpected. And so with that, I think we're very open to thinking about different mechanisms. TR beta is interesting just because if you look at some of the underlying biology, I don't want to go into too specifics here, but TR beta is it's a metabolically driven methodology of how it works. It actually increases bile acid production, CYP7A1, FXRs do actually the exact opposite. So I think the increase of bile acids is deleterious. TR betas have shown some efficacy on NASH resolution, but the increasing of bile acids in the setting of NASH is not something you want to do. And so mechanistically, it makes sense to combine the two different mechanisms, as well as the fact that FXRs do tend to have an on-target LDL effect, and clearly the TR betas are able to have a positive effect on lipids. So I think that makes sense to do. It's just a matter of whether or not those companies who have those programs are willing to do combination studies. I think GLP-1, I think we'll wait for the data come May, right? But I think that's an interesting area because of the dramatic metabolic effects that you can see that I think would also be complementary. So I'm not just saying that just because Tomas is immediate to my right, but I really do believe that it's not just one thing that you can do, and I think we'd be open to testing a whole host of different combinations because ultimately this is a complex disease where monotherapy is not going to be the way to treat it.
Thank you. And Bernard, similar combination question for you. Would you rather combine PLN 1474 with Novartis' Trifexevir, which is an FXR agonist in phase 2 development, or inclisiran, a PCSK9 siRNA that was completed phase 3 testing and was recently acquired by Novartis? Explain your answer, especially we know that NASH patients' leading cause of death is cardiovascular related.
So very similar to, I think, to Ken's answer is like, it's not up to me to kind of discuss the combinations that Novartis wants to test. I think from a high-level perspective, we have a mechanism of action which is clearly differentiated and combinable with any of those. We have a drug that has, at least based on the preclinical data, no drug-drug interaction potential, although, an siRNA is a different story. I think, again, I don't want to comment on Novartis' strategy moving forward with our compound, but if you look at what they've been doing and what is kind of the trend in big pharma right now is try everything, right? I think Novartis has a number of those joint ventures with other big pharma companies and smaller ones, and just try to, they're trying out different combinations, and I wouldn't be surprised if that's what they're going to do with our compound as well.
Thank you. Tomas, would you rather acquire a preclinical asset with a novel antifibrotic MOA that comes with a large data package but is delayed in development, or an antifibrotic MOA that's advanced in clinical development that has modest efficacy, maybe some light safety liability?
Light safety?
Exactly.
We haven't seen anything in the antifibrotic space in the late stage studies that is really making me thrilled yet. So I would go for the preclinical. Right now, the readout of the data will also guide me if there is what is the need. Maybe a little bit softer touch on the antifibrotic is enough to make me go for a later stage one, or we. So, but I would like to comment upon the issue is really preparing the liver, the fibrotic liver, for regeneration. That's the way we look at it, is that this is not just antifibrotic. You have to have components in there permitting cell migration and permitting this regenerative need that a fibrotic liver has. And that is what is guiding us in mechanism of action is really towards that end. We don't think that just solving the fibrosis, and this has been said many times, is enough, and Ken very well explained it here in the beginning of the discussion. And I think that's absolutely key. Did that answer your questions too?
Yeah, no, you did. You pick one. You picked preclinical. So you were the one with the most straightforward answer. The other ones, good job.
I'm very collaborative.
Yes, we like that. I'm going to open it up for the audience for questions. Do we have a mic, or I can walk with the mic and just pass it on? I'll be also the runner.
Hi, Scott Harris, Altimmune. Thomas, thanks for the information on semaglutide. We're all anticipating the readout on that study. We know you have other compounds coming up in the future. Could you comment on your emerging dual agonist for NASH and obesity?
Well, a lot of it is not in the public domain. So our strategy is really what we are doing in-house is metabolic. We have seen, we and others have seen very good positive data in addressing insulin resistance, not necessarily with the PPARs because of the safety, but, there are studies ongoing showing, and we will have readouts on PPARs with different profiles. So we strongly believe that the metabolic aspects of NASH will play a role, and our internal pipeline, the preclinical pipeline, is reflecting that with different types of mechanism of action. And then we also have a set and an effort on anti-inflammatory, because that's a space we've been in before at Novo, so we're very well equipped to do that in-house as well. What we don't have is, of course, anti-fibrotic. And that's really what I can say. We are completely agnostic to the modality. We do small molecules, silencing. We do, not only peptides and proteins.
Any other questions? Don't be shy. This is the time. Okay, I'm going to ask, if there's no question, one last question, which is, how set in stone is the regulatory development path in NASH? There is one. Oh, okay.
I mean, I could just kind of shout, since it's late in the hour. Can you comment about sort of biomarkers and diagnostic strategies, and sort of how do you work through that? Are you looking to outsource that to diagnostic partners or the internal biomarker programs, or how do you see that happening so that patients and doctors can have access to these important tools to make these kinds of decisions? It will be, I assume, important for your therapeutics, as we know, regimens to be defined.
Yeah, I think we need, I think everyone talks about this. Biopsy is not going to be the way that you're going to trace a patient as to whether or not they're responding. And so you're going to need something else to be able to follow. I'd say that as a therapeutics company, especially in emerging biotech, it's too much to sort of take on to think that you're going to develop your own biomarker strategy. I actually used to run a diagnostics company, so I've been on both sides of the fence. So I think it's unrealistic to think that a company can do both. But there are other companies out there that are developing diagnostics and biomarkers, whether they're looking at proteases or markers or things like that. And I think as a drug developer, we're very open to evaluating those. And so what I would sort of recommend is to anyone that's trying to develop a biomarker that could be non-invasive is that they partner and they reach out to the biotech companies, because we collect the blood. That's easy enough for us to do. It's easy enough to send over the sample of whatever it is to be analyzed. But realize that in diagnostic development, because they did this, is that that's part of your development cost. And so don't expect that you should be compensated or paid for this, because that's exploratory. And that's really going to be the best way to really drive innovation forward. I don't think there's anything yet out there that's reliable enough to say that you can track monitoring. But I think, at least from our purview, we'll be reliant upon diagnostic companies that are focused on that. And hopefully, they can just be proactive in reaching out to those that are doing the studies, because the hardest thing to obtain is patient clinical samples to validate.
I think part of our strategy, in collaboration with Novartis, is defining biomarkers, at least that are related to your specific target. I mean, biomarkers for outcome of disease is a whole different game. But at least you have your own target. In our case, it's an integrin. And so as part of that, based on what we did in IPF, is we developed a imaging agent, a PET ligand, which binds specifically to the target. So at least you can assess how much of that target is overexpressed in liver fibrosis, for example. And if you give a drug, does it really bind to that target? Because that's the start of drug development. So at least we can guide that. Whether that can be used in practice, once a drug is approved, is a totally different story. But at least we should get our act together in terms of, targeting our treatment and measure what we do.
Yeah, I agree with that. I would add to that. I mean, biomarkers are a big term. I think in the field, it's recognized a huge unmet need is these non-invasive techniques to stage patients, to measure outcomes. And there are big international consortiums working on that. And I think we're committed to working with those organizations and using our data to help them. Similar to Pliant, when you think then about our drug development, we're also developing tools. We've developed novel biomarkers for alpha beta 3. We're also looking at imaging techniques. There's clinically staged PET ligands for alpha beta 3 and alpha beta 6. So we're using those to help drug development, make sure we're bringing forward the right drugs for patients. So I think that can help make sure we're putting the dollars in the right places and using those wisely so we're dosing patients correctly, but then also contributing to the body of knowledge to help eventually non-invasive panels to identify patients, stage patients, diagnose patients, measure treatment, identify which treatments. That's going to be an enormous effort, and that's why there's these big consortiums, and no company can do it on their own.
I do think, though, that the innovation on a biomarker has to come from an emerging diagnostics company. If we rely upon the consortium, we'll be here 20 years from now still waiting. So that's my own experience.
Certainly agree. We are part of all the large consortia at Novo Nordisk, and we see the need, but it must be a biotech that drives this. And we are not a diagnostic company. It's a completely different business, as we Ken said. But the need is there. We support all the efforts, and we are taking active part in the industry consortia.
Thank you so much. Please take the time to thank our amazing panelists for taking time out of their extremely busy JP Morgan schedule to be part of this panel. So thank you so much for being here. And well done.